Somewhere in Africa, right now, there are two people standing a few hundred miles apart. They have the same skin tone, the same continent, the same, in most people's eyes, race. But genetically, they are more different from each other than a Norwegian is from a Japanese person. How is that possible? The answer to that question is about to unravel everything you were taught about race, ancestry, and where you actually come from. And it starts 300,000 years ago in a place scientists didn't even know existed until a few years ago. For most of human history, we sorted each other the easy way. By eye, by skin, by the shape of a face or the texture of hair. It felt obvious, almost automatic, the same way it feels obvious that the sun moves across the sky instead of the earth spinning underneath it. And for a couple of centuries, science went along with that instinct. In the early 1800s, an American doctor named Samuel Morton built an entire career on it. He collected human skulls, hundreds of them, gathered from battlefields, from graveyards, from wherever he could get them without asking too many questions. He would pack each skull with seeds, and later with lead shot, then pour the material back out and measure the volume. Bigger skull, he believed, meant bigger brain, and bigger brain meant a more advanced race. He ranked whole populations of human beings this way, using nothing but seeds and a scale, and for decades his numbers were treated as science. Morton wasn't working in bad faith exactly. He simply didn't have the tools to know better. He was measuring bone before anyone understood evolution, long before anyone had the faintest idea what a gene was, let alone a way to read one. So he built a hierarchy out of guesswork and called it biology. And that hierarchy didn't just sit quietly in a museum drawer, it got used. It shaped laws, justified slavery, and gave a scientific-sounding excuse to some of the ugliest chapters in modern history. The idea that race was a fixed biological fact, written into the body the same way height or eye color is written into the body, became so deeply embedded in Western thought that almost nobody thought to question it. Then came DNA. When scientists finally learned to read the actual code inside our cells, the 4 letters that spell out every instruction for building a human being, they expected, at least some of them did, to find Morton's hierarchy hiding somewhere in the genome. Distinct genetic signatures for distinct races, clean boundaries, a biological fingerprint for whiteness, another for blackness, another for every category humans had invented to sort each other. What they found instead was almost the opposite of what they were looking for. In the year 2000, the first rough draft of the complete human genome was announced at a ceremony in the White House. It was a genuinely historic moment, the kind of achievement that gets compared to landing on the moon. And one of the scientists who helped make it happen, a researcher named Craig Venter, stood up and said something that quietly detonated underneath the entire concept of race, as science had understood it. He said the idea of race has no genetic or scientific basis. Not less basis than people assumed, not a weaker basis. No basis at all. That statement sounds shocking, but the data behind it is even stranger, because it isn't just that race lacks a genetic basis. It's that the genome actively contradicts the racial map we've all been carrying around in our heads. Here's where it gets strange, and here's where those 2 people in Africa come back into the story. If race really were a biological category, the way species or subspecies are biological categories, you'd expect the biggest genetic gaps on Earth to sit between the groups we've historically called different races. Europeans versus Africans. Asians versus Europeans. That's the picture Morton was chasing with his skulls, and it's the picture most people still carry around without ever examining it. But when geneticists actually mapped where the deepest splits in the human family tree sit, that picture fell apart completely. The deepest splits on Earth, the oldest branches in the entire human family, aren't between what we'd call different races at all. They're between different populations within Africa itself. Groups like the Khoisan people of southern Africa and the Pygmy peoples of Central Africa split off from the rest of humanity, and from each other tens of thousands of years before anyone left the continent at all. These groups have been distinct, isolated lineages for so long that the genetic distance between them dwarfs the distance between, say, a Han Chinese person and a Scandinavian. Sit with that for a second, because it inverts everything the old racial hierarchy assumed. Under the traditional picture, African is treated as one branch of the tree, sitting alongside European and Asian as 3 roughly equal limbs. But the real tree doesn't look like that. It looks more like this. Almost the entire non-African world, every European, every Asian, every indigenous American, every Pacific Islander, descends from one relatively narrow branch that split off from African populations somewhere around 50 to 70,000 years ago, and then fanned out across the planet. Meanwhile, inside Africa itself, there are multiple ancient branches that have been separate for far, far longer than that. The Khoisan lineage alone may represent a split that goes back somewhere in the neighborhood of 100,000 years or more, based on genetic dating methods. Which means the 2 people standing a few hundred miles apart in Africa, both of whom the outside world would casually lump into a single racial category, can genuinely carry more distinct, more ancient, more deeply diverged genetic histories between them than an entire continent's worth of people who left Africa together and spread out into Europe and Asia. The category African turns out to be doing something almost backwards from what people assume. It isn't the narrowest, most tightly related group on the family tree. In genetic terms, it's the widest, oldest, most internally diverse branch there is, and everyone else on Earth is a smaller, younger twig, growing out of just one part of it. Let that reframe something for you. In a very real sense, every single human being alive today is African. Not culturally, not symbolically, but literally in terms of ancestry. Homo sapiens, as a species, emerged somewhere on the African continent, and current fossil evidence, including a remarkable find from Morocco, pushes the earliest anatomically modern human features back as far as 300,000 years. For roughly 200,000 of those years, while modern humans were already walking the earth, we stayed almost entirely within Africa. And even during that long stretch, long before anyone crossed into the Middle East or Europe or Asia, groups inside Africa were already splitting off from each other, migrating to different regions of the continent and becoming isolated. Those internal African splits are the oldest branches on the human family tree, and they happened long before race, in the modern sense, existed as a concept anyone could have applied. So if the deepest, oldest divisions in humanity aren't between the races we talk about today, where did those categories even come from? The honest answer is that they came from geography, recent geography, and from the way small groups of people who left Africa later became isolated from each other for comparatively short stretches of time, geologically speaking. When a small population migrates into a new environment and stays cut off from other groups for thousands of years, random mutations accumulate. Some of those mutations affect visible traits, skin tone shifting to match local sunlight levels, eye shape, hair texture, bone structure. Over generations, those visible differences become the traits people use to sort each other into categories. But visible traits are a tiny, almost trivial slice of the genome. They're the equivalent of judging two books by the color of their covers, while ignoring that one is 400 pages longer, written in a different structure, and contains entire chapters the other one doesn't have. This is really the heart of what the Human Genome Project revealed when its first phase was completed. Human beings, on average, are about 99.9% identical to each other at the DNA level, regardless of what continent their ancestors are from. That's an almost absurdly small margin of difference, and it means there's more genetic variation within what we casually call a single race than there is between different races. Two random people who'd both be labeled white in a census can be more genetically different from one another than one of them is from someone labeled Asian. Skin color and a handful of other visible traits are controlled by a small number of genes that happen to respond quickly to environmental pressure like sunlight exposure. But those genes are a rounding error compared to the full genome, and they don't correlate cleanly with the deeper, slower moving patterns of ancestry that actually tell the story of human migration. Formally, biologists have a word for genetically distinct populations within a species. It's subspecies. Wolves have subspecies. Tigers have subspecies. Plenty of animals do. But when researchers went looking for that kind of clean, physically and genetically distinguishable subdivision within Homo sapiens, they didn't find one. As a species, we don't split into subspecies. Every human population blends into every neighboring population through gradual, continuous variation, not sharp genetic boundaries. There's no line on the map where one race's DNA stops and another's begins, because that line was never there to begin with. What's there instead is a gradient, a slow smear of genetic variation, thickest and richest in Africa, thinning out and becoming more uniform the further you trace human migration outward, because every new population founded outside Africa started from a smaller, less diverse founding group than the one it left behind. This isn't just old news from the year 2000 either. It's something researchers keep confirming, with bigger data sets and better tools, right up to the present. One of the most striking recent examples comes from a massive study using the All of Us research program, a genetic and health database backed by the National Institutes of Health that has been working to sequence genomes from a huge, diverse cross-section of people living in the United States. Researchers led by a genetic epidemiologist named Charles Rotimi analyzed roughly 2 million genetic variants across more than 230,000 participants. What they were checking was simple to describe and fascinating in its results. Does the race or ethnicity people report about themselves actually match clean, distinct genetic clusters? It didn't. Instead of splitting neatly into the racial categories people checked off on a form, most participants' genomes showed broad, overlapping gradients of ancestry that spanned multiple continents at once. People who identified as Black or African American showed wildly varying proportions of African and European genetic ancestry, not a single uniform signature. And the pattern got even more dramatic among people who declined to select a racial category at all. More than 90% of that group identified as Hispanic or Latino, and their DNA reflected a genuinely broad mixture of African, European, and Native American ancestry all at once, a mixture that no single racial checkbox could ever have captured accurately in the first place. A bioethicist who reviewed the study made a point that cuts right to the center of all this. She said, researchers shouldn't default to broad continental categories anymore, because those categories are, in her words, artificial, not slightly imprecise. Artificial, built more for administrative convenience than biological accuracy. And it's worth noting, this isn't some fringe opinion. It's become close to a consensus position among population geneticists, most of whom have quietly moved away from using the word race as a scientific label at all in recent years, replacing it with more precise language about ancestry, migration history, and regional genetic variation. Now, none of this means self-reported race is meaningless as a concept. That's an important nuance, and it would be dishonest to skip past it. Race is real as a social category, real in the sense that it shapes how people are treated, what neighborhoods they grow up in, what kind of health care they receive, what kind of discrimination they face. Those social realities leave genuine fingerprints on health outcomes, on stress levels, on access to resources, and researchers studying disease and public health still find self-reported race useful precisely because it captures those social and environmental factors, things that pure genetic ancestry data can't see on its own. The mistake isn't in acknowledging that race shapes people's lived experience. The mistake is assuming that lived experience maps cleanly onto a fixed, ancient biological category, when the genome itself tells a much messier, much more interesting story. And that story gets even stranger once you zoom out from modern humans entirely and start looking at our deeper past. Because it turns out the version of human history most of us grew up with, one species, evolving alone, spreading out to fill the planet, isn't quite right either. For most of the time that anatomically modern humans have existed on Earth, we were not alone. We shared the planet with other species of humans, ones with their own tools, their own migrations, their own family lines. It's only in roughly the last 30,000 years, a genuine blink of an eye on an evolutionary timescale, that Homo sapiens has been the sole surviving human species on the planet. Before that, for a period stretching across hundreds of thousands of years, several different kinds of humans coexisted, sometimes overlapping in the same regions, sometimes even in the same caves. The first real proof of this came in 2009, when scientists successfully extracted enough genetic material from ancient Neanderthal bone to sequence a full genome from a human species other than our own. That sequence answered a question that had haunted paleontologists for generations. Did modern humans and Neanderthals ever actually interbreed when their paths crossed? The genome gave a clear, unambiguous answer. Yes, Neanderthal DNA carries traces of Homo sapiens DNA, and Homo sapiens DNA carries traces of Neanderthal DNA. On average, people of European descent carry somewhere between 1% and 2% Neanderthal ancestry sitting quietly inside their genome right now, a genetic souvenir from encounters that happened tens of thousands of years ago. Then just a year later, the story got even more surreal. In a cave in Russia called Denisova, researchers found a tiny fragment of bone, the tip of a finger, and a single molar tooth, both belonging to a teenage girl who had died there long ago. That was enough to extract a full genome, and when scientists compared it to both modern humans and Neanderthals, it didn't match either. It was something else entirely, a previously unknown branch of the human family tree, now called the Denisovans. Like the Neanderthals, they interbred with our ancestors too, and the genetic evidence for that mixing is written into the DNA of people alive right now. Interestingly, the geographic pattern of that ancestry isn't uniform. The further east you travel across Asia and into the Pacific, the more Denisovan ancestry shows up in modern populations, and the less Neanderthal ancestry appears, a genetic echo of exactly where and when those ancient encounters happened. But here's the detail that really rewrites the picture, the one that even surprised the researchers studying it. When scientists compared how much Neanderthal and Denisovan DNA modern humans actually carry, the numbers didn't quite add up. There was more genetic variation floating around in the human genome than could be explained by mixing with just those two known groups. That discrepancy points to something unsettling and genuinely thrilling at the same time. The likely existence of at least one more archaic human species that we interbred with, one for which we currently have no bones, no fossils, no physical trace at all. Its only evidence is a shadow, a statistical ghost, hiding inside the DNA of people alive today. We don't know what it looked like. We don't know its name because it never got one. But it's in us anyway, an entire lost species surviving only as scattered fragments spread across the genomes of its distant hybrid descendants. Geneticists studying this kind of hidden history sometimes call their work genetic archaeology, because that's essentially what it is. Instead of digging through dirt to find bones and tools, they dig through the genome itself to find traces of populations that left no other record. Researchers doing this kind of work have pointed out that even after sequencing something like 100,000 human genomes and building increasingly sophisticated tools to analyze that variation, the deeper they dig, the more hidden stories they keep finding buried inside our DNA. Every new dataset seems to reveal another layer of mixing, another lost population, another migration nobody had documented, another piece of evidence that human history was never a single clean line moving from point A to point B. It was always a tangle. This tangle also shows up in more recent, more subtle ways, not just through ancient interbreeding, but through the environments different populations settled into after leaving Africa. When farming emerged around 10 to 12,000 years ago, it didn't just change what humans ate. It changed human biology itself. The clearest example is something called lactase persistence, the ability to comfortably digest milk sugar into adulthood. Most mammals, humans included, historically lose that ability after weaning. But in populations that domesticated dairy animals and built milk into their diet over many generations, a genetic mutation allowing adults to keep digesting milk spread rapidly because it offered a real survival advantage. Today, most people of European descent carry that mutation, while most people worldwide still don't, a difference that has nothing to do with any ancient racial hierarchy, and everything to do with which specific population happened to start herding cattle and drinking milk a few thousand years ago. There's an even more extreme, more recent example of how fast the human genome can respond to a specific environment. On an Indonesian island near a cave, where the remains of a separate extinct human species called Homo floresiensis were discovered, a population of modern humans living nearby also evolved unusually short stature, apparently completely independently, unrelated to the ancient species that once lived in that same cave. It's a striking coincidence, two entirely different human lineages, separated by tens of thousands of years, both landing on small body size as an adaptation to the exact same island environment. A reminder that evolution keeps finding similar solutions to similar pressures over and over, regardless of ancestry or what people would casually call race. So where does all of this leave the old idea, the one Samuel Morton was chasing with his skulls full of seeds, the one that assumed humanity split cleanly into a handful of fixed biological races with their own separate destinies? It leaves it in pieces, and not because modern scientists set out with an agenda to dismantle it. It fell apart simply because the actual data never supported it in the first place. The deepest splits in humanity sit inside Africa, not between Africa and everywhere else. The most common ancestry in every living human being traces back to that same African origin. The visible traits people have used for centuries to sort each other into races represent a tiny, fast-changing sliver of the genome, while the vast majority of human DNA doesn't sort along those lines at all. And when researchers directly tested whether self-reported race matches distinct genetic clusters in a massive modern real-world dataset, it didn't. Ancestry turned out to be a gradient, not a set of boxes, a smear of shared and overlapping histories rather than a handful of separate branches. What replaces the old racial map, if you're looking for a better mental picture, is something closer to a river system than a family tree with neat, separate limbs. Picture countless tributaries branching, rejoining, splitting again, some flowing together for a while before diverging once more, others merging back in after tens of thousands of years apart. Neanderthal water flows into that river. Denisovan water flows into it too. So does the genetic contribution of at least one more ghost population we haven't even identified yet. Every person alive today is a unique confluence point in that river system, carrying traces of migrations, isolations, reunions, and adaptations stretching back 300,000 years, almost none of which lines up with the handful of simple categories a 19th century skull collector once tried to sort humanity into using pepper seeds and a scale. It's worth pausing here to ask a slightly uncomfortable question. If the genetic evidence has been pointing this direction for over 2 decades now, why does the old racial map still feel so intuitive to so many people? Part of the answer is simply that vision is a shortcut, and human brains love shortcuts. Skin tone, hair texture, and facial structure are the first things we notice about a stranger, so it's natural that early scientists, working centuries before anyone could read a single gene, built their entire framework around the traits that were easiest to see. The problem is that easy to see and biologically important turned out to be two very different things. The genes controlling skin pigmentation are a strikingly small slice of the genome, and they happen to respond quickly to environmental pressure like sunlight, which is exactly why they change so fast across geography and why they're so visually obvious. But fast changing and visually obvious doesn't mean deeply important to the underlying structure of human ancestry. It just means visible. A handful of genes reacting to sunlight over a few thousand years can completely redraw what a population looks like on the outside, while barely touching the deeper architecture of who that population is actually related to. There's also a historical reason the old framework stuck around so stubbornly, and it isn't really about science at all. Once a racial hierarchy gets built into laws, into property rights, into census categories, it stops behaving like a hypothesis that can be quietly revised when new evidence shows up. It becomes infrastructure. Entire legal and economic systems were constructed on top of Morton's skull rankings. And infrastructure is slow to dismantle even after the foundation underneath it has been proven hollow. The categories persisted, not because the genetics supported them, but because so much else had already been built assuming they were true. Modern researchers studying ancient DNA keep finding more evidence that complicates the old picture even further. Take the discovery, made through analysis of ancient remains, that a benign bone tumor existed in the rib of a young Neanderthal who lived roughly 120,000 years ago, by far the earliest tumor ever identified anywhere in the archaeological record. It's a small detail, almost a footnote, but it's the kind of thing only this new generation of research can reveal. A tiny fragment of individual medical history preserved for over 100 millennia and only readable now because of how far genetic and archaeological methods have advanced. Details like that are a reminder that the story being told through ancient genomes isn't an abstract statistical exercise. It's built out of real individuals, real lives, real bodies that existed long before anyone had invented the categories we now argue about. Consider, too, just how much of the human genome remains genuinely unexplained even today. Scientists studying complex human traits, height, blood pressure, disease susceptibility, have run into what's called the missing heritability problem, where genetic factors clearly influence a trait, but researchers can't fully account for where that influence actually comes from. Some recent work suggests the missing piece isn't that heritability doesn't exist, but that existing tools simply haven't been sophisticated enough to find it yet, hidden in interactions between multiple genes, rather than sitting in any single obvious location. That's a humbling reminder for anyone tempted to think the genome has been fully mapped and understood. We've sequenced an enormous number of genomes at this point, and the tools for analyzing that variation keep improving, yet researchers openly admit that the deeper they look, the more hidden stories keep surfacing. Stories about migration, mixing, and quiet ancestral threads nobody had documented before. All of this adds up to a genome that behaves less like a filing cabinet with labeled folders for each race, and more like a layered archive, one where every new tool for reading it uncovers another buried layer nobody expected to find. Ancient interbreeding events, ghost populations with no fossil. Record at all. Independent evolutionary experiments like short stature evolving twice on the same island for two completely different reasons. Environmental adaptations like lactase persistence, spreading through specific populations because of what they happen to eat. None of these threads sort neatly into racial boxes because the underlying reality was never boxed in the first place. It was always continuous, always blended, shaped far more by geography and time than by any fixed biological category. And this reframing matters for reasons that go well beyond satisfying curiosity. Medicine has spent decades sorting patients into racial categories on the assumption that race itself was a reliable stand-in for shared biology, using it to estimate everything from kidney function to drug dosages. But researchers who have reviewed how African ancestry gets used across hundreds of published genetic studies have pointed out a quiet, persistent confusion running through much of that work. Many studies seem to treat African ancestry as though it marks a single biological population sharing DNA unique to itself, when the actual evolutionary picture shows something closer to the opposite, a continent containing more internal genetic diversity than the rest of the world combined. That confusion isn't just an academic footnote. When a category that doesn't reliably track biology gets used as a proxy for biology in a lab, it can distort research findings and, in the worst cases, patient care. Recognizing that ancestry is a gradient rather than a set of fixed racial boxes isn't just a philosophical correction. It's already reshaping how disease risk gets studied and how treatment decisions get made. The two people standing a few hundred miles apart in Africa aren't an exception to some tidy genetic rule. They're a preview of what the entire genome actually looks like, once you stop assuming race is written into our DNA and start asking what's really there instead. And what's really there is bigger, older, stranger, and far more interconnected than any map of races ever managed to capture.